Hi guys, Dr. Borthwick here. Thought I'd just give you a wee intro starter to your assignment this week. So we're looking at protein structure. Now last time we looked at amino acid structure. We're going to continue with that and then look at the other three levels of protein structure: secondary, tertiary and quaternary. All the learning outcomes are here, some overlap from last time. So heads up, obviously talked about amino acids last time. You do not need to recognise every amino acid. You need to recognise the structure of amino acid and that it's which group it belongs to. Is it acidic, alkali, ionic or hydrophobic? Okay, so a little bit of this is from higher chemistry. For those who've done that last year, you'll be okay. Those who don't worry. I'll try and I've explained it as reasonably simple as possible. So, when you've got your amino acids, they need to join together to give you your polypeptide. So your polypeptide is made when amino acids join together in something called a condensation reaction. What's that? Well, think about windows. And you get condensation in windows, water is produced. So if you look for the carbon with the double bond oxygen and the nitrogen there, what happens is water comes out there and that bond then forms between the carbon and the nitrogen. So that is called your peptide bond or peptide link. In chemistry, we called it the amide link, but it's biology now, so we're going to call it the peptide link. Okay, so we've got chains of amino acids. This forms on that. So you've got lots and lots of amino acids joined together to form a polypeptide. Proteins are then made up of different structures within these polypeptides. So there's three different structures that can form: alpha helixes, parallel or antiparallel beta sheets, and turns. So alpha helix, you might have come across DNA. It's got this lovely sort of idea that you've got a helix like this. How are these held together? They're held together with something called hydrogen bonding between the hydrogen of the peptide bond and the oxygen of another one. So down here you can see it all being held together. Okay, that's how your DNA is held together. In this case, it's usually just, your proteins are held like that as well. So beta sheets, you've got two different kind of beta sheets. That should disappear. Well, you've got your beta sheets here. So they've got antiparallel. So this is your polypeptide coming along here, amino acid sequence, turns, goes along the other way, and then it would turn and go along the other way. So that's called antiparallel. Whereas if you've got the parallel one, get rid of this. What happens is your beta sheet's going along here. It's a big, huge turn, and it means the parallel goes along there and it carries on. It's also held together by these intermolecular forces. And then the final secondary structure is a turn. So a turn is this, like that. It could be within your beta sheet. It can be from one alpha helix to another alpha helix, or a beta sheet to a beta sheet. Okay. So when you've got lots of those going on, for instance, here you've got some beta sheets, you've got some alpha helixes there. It's all held together, and this is now called the tertiary level or structure. And how are all them kept held together? By 5 different kind of interactions or intermolecular forces. Okay, so just quickly go through each one. Hydrophobic is you've heard of that before. Hydrophobic, obviously, like hydrophobic with like hydrophobic will hold the bonds together. Hydrophilic on the outside. So membrane proteins, which we'll look at after the summer. A lot of how they're held in the membrane is because the hydrophobic is like with like. So membrane's hydrophobic, protein part is hydrophobic. Ionic bonds, that's with, obviously, learned about that last time about the different R groups like arginine and aspartic acid. Positive and negative, opposites attract. A lot of the acidic alkali ones are like that as well. So ionic bonding is a little bit like hydrogen bonding as well. As you're doing chemistry, and this is like the difference between, they're both sort of what we call polar bonds, but in this case in biology they want to call them ionic bonds. So chemists, polar bonds and ionic bonds are the same thing. So you've got your polar-polar interactions and you've got your hydrogen bonding. Hydrogen bonding is a special kind of ionic bonding in this case, between the hydrogen and the oxygen, and hydrogen and nitrogen. Okay. London dispersing forces are the weakest kind of intermolecular forces. They can be explained by every atom in the molecule has electrons. They're flying around the centre nucleus all the time. They might all go to one side, whereas a neighbouring atom, they might all go to the other side. If this happens, there is a very fleeting bond, which is called a London dispersing force. Finally, something new for everybody is called disulfide bridges. They're formed when you've got your R group of things like, cysteine, amino acid, and it forms, it's a covalent bond that's formed between the two sulfurs. And that's quite a strong bond there. So the final one is quaternary structure. So you've got the whole protein now. You've got several polypeptides that are all held together. How are they all held together? They might be held together with all of the previous kind of bonding that we just talked about. And they might hold together a ligand. So a ligand is something extra, like it could be an ion, it could be another protein that's held together within the protein. So for instance, chlorine has magnesium hold on there, your heme group has iron stuck in there, and various other things. So the next few slides are just basically examples of them. This should be not new to you, but denaturation with temperature and pH, so have a look at that. Now the final thing I just wanted to quickly look at is this web page here. So I've given you a worksheet to do. First lot of questions are just general protein questions. Second lot are all to do with this website. So you click on that website either on here or on the worksheet, you get to this website here. Now I do apologise, the first thing that comes up with is obviously coronavirus. You want to investigate something, so you could go onto the sheet and look, right, okay, I need to investigate this. So you could just do the whole word, or you could just copy and paste. I've given you instructions about how to do that as well. They're slightly old, so they might not be quite up to date. You go onto there, and we don't want that bracket. That doesn't matter, it's come up. So ask for that one. And you want to look at the structure here. So if you want to take more time to go look through the structure here, I realise I'm sort of going over here quite a lot, but this is quite fun. So you can go on here, you find all the information, click on that, and you can start moving this around. You can see alpha helixes, beta sheets, and turns. Okay, so go through the worksheet and you can answer the questions on them. Give me a shout if there's anything you don't understand. Thanks. Bye.